So welcome everybody. Thanks again for joining us in Apiculture Online, Hive Chat with NC State. My name is David Tarpey. I'm a professor here at North Carolina State University. As always, we're joined by members of our apiculture program, as well as the apiary inspectors of the North Carolina Department of Agriculture. So thank you again for joining us tonight. We have a great lineup tonight. Our special guest is Dr. Kaira Wagoner from University of North Carolina at Greensboro. She's going to be talking a little bit about her research and some of the things that she's been doing there. But first, we're just going to go ahead and launch into some quick announcements of what's going on in the upcoming months. I just want to make sure that everybody knows of and is reminded of our most recent offering of our Bees Intermediate Academy. This is something that we did in 2019, but in 2020 we kind of put it on hiatus because of COVID. But we're relaunching this. It's a 2-day intensive course for not beginner beekeepers, but not really advanced beekeepers either. So it's a full 2-day. It's a Saturday, Sunday, so it's a weekend only at the Union County Extension Center down in Monroe. So it's very centrally located for both North Carolina and South Carolina as well. But really anybody's welcome. So feel free to find out more information on our website and to register today. It's a really great way to take your beekeeping to the next level. If you've been keeping bees for just a year or two, we like to think of this as a booster shot of your beginner bee school, where we tend to go into a lot of the same topics, as well as additional topics that you might have learned in your bee school. But now that you have some practice and some practical experience in bees, we go into a little more depth, and hopefully it resonates a lot more in trying to understand the bees and beekeeping. So these have been extremely successful in the past. So really encourage any and all of you to consider this. But first, as always, our first segment in Apiculture Online that we do every single week is Bees in Season. And this is in essence what your bees are doing right now and what you should be doing as a beekeeper in order to assist them. And so right now, as of the end of July, right, we're just kind of in the height of summer. Summer has definitely arrived. Earlier this season, it seemed like it might not ever occur. It was the second wettest winter, at least in the Piedmont of North Carolina, on record, and then the driest spring. And it was also fairly cold, or at least it didn't really warm up, or it kind of lagged for quite a long time. But the summer heat is finally here. The spring nectar flow is long gone. So, the downhill sledding of beekeeping season is now over. Now it's this kind of tough uphill climb where the bees are kind of nutritionally deprived. There's not a lot of natural flow out there. It's really, really hot for them. So they're spending a lot of time cooling the hive down. This is the height of robbing season, which we'll talk about in just a moment. So it's definitely things that you need to look out for. But as usual, 75% of the problems that beekeepers deal with starting this time of year and throughout the summer and into the fall is nutrition, queen problems, and of course, varroa mites. These are the main things that if you can have a good management plan for all three of those things, you're going to have an incredibly much higher likelihood of success in your beekeeping. Queen problems are often associated with swarming and things kind of earlier in the spring. You're not going to really see a lot of swarming this time of year, but what you can start to see crop up this time of year is supersedure, which is the old mother queen gets replaced. Rather than being replaced after swarming and splitting, they just replace her while she's still there. And so it can be kind of cryptic, and you might not realize it, but when you go into your colonies and you see queen cells this time of year, it's probably not because they're swarming. It's probably because they're trying to replace that queen. That's usually an indication, not necessarily that there's something wrong with the queen, but there's something going on with the colony overall. So you need to be very vigilant and and try to drill down and try and diagnose that. Nutrition as an issue is just it. Usually there's a lot of pollen flow going on even in the middle of the summer. Bees are able to forage for pollen that's out there, but it's the nectar, the lack of nectar flow that really makes the bees hungry, grumpy, stingy, the whole bit. So, and bees are so much happier when they have a natural nectar flow. You can provide them sugar syrup, and yeah, they might take that down, but it's just not quite the same. So, hive location during the middle of the summer can be really, really difficult because there's not a lot of natural bloom out there, but it really is really quite important. And of course, varroa mites. Aaron did a great job last month in our last Apiculture Online talking about how to count for mites, how to monitor for mites. We have other recordings on this in previous Apiculture Online recordings on our YouTube channel, so go and check those out. Next month, we're going to have a special guest from the USDA talking about varroa mites. So we'll be talking about mites further in this session in our series here, because they really are so important, especially this time of year. So if you haven't already started monitoring for mites, you got to go out and do it right away. You got to start doing it right now if you haven't already, because right now is really very much the time when all these other stresses impact the colony and the mites can really start taking off. And if you don't do anything right now to control them and keep those populations down, they're going to be in big trouble come late summer and fall. Because of the nutritional problems that are going on, lack of nectar flow, even in the mountains. We were talking earlier offline that even the sourwood flow up in the mountains of North Carolina and Appalachia, that's pretty much over as well. And so there's a real nectar dearth. And so, honeybees, they like to make honey, but why make it when you can steal it? And so, honeybees are really good at parasitizing and robbing out honey from weaker colonies. And so if you have multiple hives within your yard, or you have some colonies that are really large or versus really small, you want to protect those small colonies from getting robbed out by the larger colonies. If you only have small colonies in your yard, there might be hives from other places that might come in and rob out your colonies. So don't think that just because you don't have any really large colonies that you're safe from robbing. No, robbing is always going to be a problem for smaller colonies that are not able to defend themselves. So installing robbing screens this time of year is really paramount. And so put them on if you haven't done so, because the robbing itself, not only is it, kill off your weaker colonies, which you obviously don't necessarily want, but it's a great way to spread disease and mites and all of these other problems. And so you don't want to contribute to that. And so putting robber screens on is really, really important. There are many different designs of these robber screens that are out there. This is just one of many examples. So, just do a Google or go into some of the beekeeping trade magazines or looking at some of the suppliers for the robbing screens. But in essence, the principle is fairly simple. Rather than having a full entrance open, which for smaller colonies might be hard for them to have enough guard bees to guard the entrance from foreign, would-be robber bees. What they do is they have a screen in front so that the hive odors emanate from that entire entrance, but it's screened off and there's only one or maybe a couple smaller entrances for the bees of that hive to come in and out. And so since they're in that hive, they know where the entrance is. Because it's smaller, it's easier to defend. And so even smaller colonies can defend a small opening like that versus a large one. But the robber bees don't know the difference. They don't belong to that colony. So they just get the hive odors from the entire entrance. And they're trying to get in everywhere, but it's screened off, so they can't. And then the bees from that hive, they are able to freely, fly in and out of the entrance because they know where the entrance is. So it's quite ingenious, and it's something that works extremely well. These can also double as moving screens where you can shut off the entire thing if you need to pick up your hive and move it to another location that might have a better nectar flow this time of year. It's something that, you know, you may strongly want to consider, that your backyard or wherever your apiary is was very convenient, but it just doesn't have very good nutrition during the middle of the summer. You might want to think about finding another location that's located near, you know, something that's blooming in an agricultural field or in a forest or something like that, so that they can have some more natural nectar flow during this difficult time of the season. So anyway, something to consider, those robber screens. Now, the main thing that we need to do as beekeepers is to help the bees help themselves. So what we should always be doing is thinking 1 to 2 brood cycles ahead, which is 3 to 6 weeks ahead from now. What are the bees going to be doing 1 to 2 brood cycles from now? And what can we do now to help them go towards that eventuality, right? So, you know, we need to be, again, August is going to be really, really hot. So they need good water sources. We need to help them with monitoring, controlling mites. Again, the nectar foraging or some sort of nutrition to make sure that they're able to sustain their brood rearing and everything else, and to prevent robbing. These are all things that we can do now that will help them down the road. This is also a fairly good time that you can requeen your colony. Again, if you go into the colony and you see queen cells and they're attempting to supersede the queen, there might be something wrong with her or something wrong with the colony, in which case it may be good to replace the queen yourself so that you don't lose several weeks of brood production. You have control over the genetics and the type of queen that you put into that colony. So if you're raising your own colony or your own queens, or you can get them from a reputable local source, midsummer, this time of year, can actually be a very good time to requeen, because then you have a relatively young queen going into next spring, going into the winter and coming out next spring, that are going to be less swarmy and more productive next spring. So definitely think of that. And as I was just mentioning, you might want to consider moving your hive location to take advantage of proximate nectar flows so that they're not as stressed out during the summer months. There's been some debate and discussion about, you know, moving your hives is stressful on them. And while that might be true, that pales in comparison to the stress of not having a lot of food available. So, you know, moving the hives is something that I think a lot of beekeepers are reluctant to take advantage of, but it can be one of those things that really, really can help your bees. Any other points or things that somebody would like to mention on the call? Dawn, Bridget? Anything about what's going on this time of year and what beekeepers should be doing?
Stay hydrated yourself. It's hot out there.
That's very true too. Yes. Don't forget to keep yourself cool, hydrated, and safe. Certainly. It's very, very, very true. Good point, Kara. All right. Well, then, hearing none, our timely topic for our second segment here is going to be on hive products, and especially hive products that are not honey. You know, so many beekeepers, we've gone through the major spring flow. A lot of beekeepers have already extracted their surplus honey, and, you know, they've gone through and done that. Hopefully you've left plenty for the bees to make them through, to help them make it through the dearth, as well as into the fall and through the winter. But there's lots of different hive products that beekeepers can think about above and beyond honey. And so this is just a really kind of quick discussion and an overview of some of those different products that you might want to think about and take advantage of. If you've extracted your honey and that's just kind of routine now, especially if you have surplus honey or you're not really sure what to do with it. These might be some other things that you might want to think about. And each one of them has their pros and cons, but they can be equally as rewarding as keeping your bees for honey. The first one is mead. So, if you're making way more honey than you can ever give away or eat yourself, this is a really great way to take advantage of some of that surplus honey, and that is to make fermented honey wine or mead. This is a very, very traditional alcoholic beverage that dates back to prehistoric times. And it's simply diluted honey. So it's usually 1 part honey to 4 parts water. And honey itself doesn't spoil because it's too sweet. But once you dilute it, well, then it can spoil. But spoiling here is you actually add the yeast so that the yeasts then go through and their whole process, fermentation process, right, and consume the sugars in the honey, and then produce alcohol and carbon dioxide as the byproducts, and that's the very basic, simple recipe of brewing and making these alcoholic beverages. Now, over time, mead makers have done different things to this traditional mead of just honey, water, and yeast. And they've done things like they add herbs and spices to augment the flavors of the mead, and those are known as metheglins. You can actually have more honey than that kind of 1-to-4 ratio. You can make it even sweeter, and those are like kind of dessert wine type things called sacks, and those are usually really, really excellent. Again, much sweeter than kind of normal meads. Or they can add fruit or juices or both in order to augment the flavors. Those are known as melomels. So these are different varieties of meads that people can make. And then from the melomels, by adding these other sources of sugar in addition to the honey, you can add, apples or apple juice, grapes or grape juice, even barley and malted grains. And these can make meads that are very similar to your beers, wines, and hard apple ciders. And so there's a lot of different recipes that are out there. The process is fairly straightforward. It's much more akin to beer brewing than it is to winemaking. And therefore, it takes a matter of weeks or months rather than months or years in order to make meads. And so this is something that you very well might want to consider, especially if you have way more honey than you know what to do with. It can be a really, really fun process if you want to get into it. There are other hive products, though, that you can harvest from your colonies, one of which is pollen. Now, pollen, unlike honey, where you go in and you pull out the frames, right, and you actually extract the honey from the frames themselves, the way to trap and collect pollen from your colonies is before it ever even makes it inside the hive, let alone into the comb. So what you can do is you can purchase any number of different designs of these entrance traps. So what this is just a plastic device that gets placed into the entrance of the hive. And so the only way in or out are through these very narrow slits, which are just big enough for the workers to fit through. But the problem is that as the pollen foragers, as they're coming back, it dislodges the pollen pellets off of their hind legs, and then it falls down into this trap right in front of the hive, right beneath them. And so this is a great way to just put it on, let the pollen just kind of fall off their legs and collect in the trap. And then you come, at the end of the day, and now you have, a pound or two of pollen that you're able to keep and do something with. Now, of course, you can't keep this trap on very long because then they'll become pollen depauperate and starve, and they won't be able to raise brood. So the good beekeeper goes in and puts it on one day, takes it on the next, put it on one day, takes it off the next. But in doing so, you can make pounds and pounds and pounds of corbicular pollen that you can use for all sorts of purposes. Now, It's been very popular for health supplement, and people eat it for protein, even though it's kind of up to debate whether or not we can extract the protein from pollen in this way. Some people even take it for allergy relief, although again, medical evidence is kind of iffy as to whether that works or not. But if you do it and it works for you, keep doing it. If you have bad allergies, you might want to try it. But the biggest benefit, I think, of trapping your own pollen, especially in the spring and summertime, is that it is a great way to then take that pollen, put it into a plastic bag, throw it into the freezer, and you can collect pounds and pounds of pollen that you can then make pollen patties, supplemental pollen patties next winter. So there, again, there's not a lot of times in the southeast of the US where there's really zero pollen coming into colonies, but sometimes it can really help to boost the brood production, especially in the early spring. And so you can take that frozen pollen from last year, mix it up into your own pollen patties, and then place them onto your colonies to give them that protein boost so that they build up all the faster in the spring. So that's definitely something to consider. And you know, you don't have to buy your own pollen patties if you just think a little bit ahead and trap your own pollen. So it's another thing that that you can do. Now, of course, wax is another popular hive product that a lot of beekeepers use and make do of. It's something that if you're going through and scraping burr comb and you know all of these extra pieces of beeswax that you just don't want, and you're throwing it on the ground, you're actually wasting a little bit of a resource. What you should do is you should take all that old burr comb, the old frames that you're recycling, so you're putting in new frames to replace some old ones. You should take all of that and not throw any of that wax away and put it in a bee tight container. You definitely don't want robbing to occur with kind of drippy burr comb or anything. You want to put it into a plastic bucket or something with a very, very tight lid so that wax moths and robber bees don't get into it. But once you have enough, you can then put it into a very simple solar wax melter. You can buy these or you can even make these. They're not very difficult design. It's just it's like a greenhouse that's on a slant, and you put there's a metal container inside where you put the beeswax, the radiant energy from the sun heats it up above 149 degrees so that it melts. It then drains down and collects into different pans. You can then filter that and sell it back to beekeeping supply companies. You can use it to make candles. You can often trade it in at some beekeeping supply companies for a discount on your wax foundation for your new frames. So it's another thing, another resource that you might be able to take advantage of. Another product that you might want to consider, although we're starting now to kind of get into a little more bizarre type products out there, you can trap propolis. Propolis, of course, is the bee glue. It's made by tree resins mixed with beeswax itself, and they use it to seal the cracks and the crevices within the hives to keep it water and practically airtight. And so what the bees really don't like are kind of cracks in their ceiling. And so they make these plastic traps that look very, very much like a queen excluder material, a plastic sheet, very thin, but none of this, unlike the queen excluder where the workers can pass, but the queen can't, these are all too small for any bees to pass. And you put that above the brood nest, you know, just below the inner cover, so you're not, you know, impeding the any bees trying to walk through the combs or anything. But what this does is it gives them cracks in the ceiling, and they go crazy because they don't want those cracks in the ceiling, and so they'll seal them all with propolis. And what you can do then, once that the propolis has been deposited in these little cracks, you can take the trap off, throw it into the freezer to get the propolis really hard, and then you can twist the plastic trap so that it all kind of breaks off and falls out of the trap. And then you can collect all of that propolis and use it for different reasons. Now, it's been used for millennia for different health applications because these resins, the tree resins, have known antimicrobial properties. So these are really, really good for wounds. They're very good for kind of dental health. They're good for scrapes and cuts, all sorts of things like that. A lot of people have used them. They put them in a tincture so they can dissolve the propolis in, kind of an alcohol or something like that. And they can put a drop or two in their tea, and it provides, health benefits that way. It's also even been used for crazy things like cancer treatments and even wood varnish, because it has that nice kind of auburn color to it. And so you can trap your propolis and use it in many different ways. One last, perhaps bizarre hive product that people can take from their beehives is royal jelly. So of course, royal jelly is that milky liquid that is provided to queen larvae as they're developing. And so the way to make quantities of royal jelly is you, in essence, cause a colony to go into queen rearing mode by removing the queen, grafting in bars of queen cells into the hive. The nurse bees will provision those larvae with royal jelly, and then you go back in 3 days later, you scoop out the larva, and then you kind of scrape out and scoop out all of the royal jelly from each queen cell like this. And for each queen cell, you get, maybe a thimbleful, maybe probably not even that much. And so, you have to do hundreds and hundreds of queen cells, and even to get kind of a measurable amount of the royal jelly. So it's very expensive. It's very difficult to harvest it, but it's something that is done. It has been, like some of these other products, used for health supplements. As far as we know, it does not turn us normal people into royalty. That has not occurred yet, but nonetheless, it can be a very popular health supplement that some people use. Now, you can buy royal jelly in quantity, like pounds of it, for maybe $100, $150, something like that. So, if you were to do that yourself, it would take forever, but you can buy a lot of it because it's commercially produced in China. And so it's very inexpensive overall for the amount of effort that it takes. But if you do use that for any sort of health supplement or whatever, that's your choice. But do not use royal jelly imported from China in your beehives, because it may have viruses or other pathogens in it that might be able to infect your bees. So make sure that you use it for kind of human purposes and not for bee purposes at all. Any other comments on hive products from those of us on the Zoom call?
David, I've got a question.
Yeah.
I was approached the other day from someone who was wanting to or considering importing foundation, beeswax foundation. My reply to him was that, as far as the state is concerned, we don't have any way of checking for pathogens like you would in the royal jelly.
Yeah.
But I don't know what the federal position would be on that. And again, I'm not trying to throw you a curve, but I don't know the answer.
Yeah, I mean, I don't know the answer either, but I do know that there have been some studies looking at how many pathogens can survive in beeswax outside of the host bees for a very, very long time. Other than American foul brood spores, which you very, very well know can persist for like ever.
Yeah.
A lot of the viruses and some of the other microbes do not survive very well in wax, especially when it's heated up, to 160 or so degrees. A lot of the commercial operations also ultra filter their wax, which is why it comes out so nice and clean. So I would probably say that that's not a huge risk compared to a lot of other things.
Right.
But
Yeah,
you
I
know,
would
when you're
agree.
importing them from other places, you know, you need to be very, very cautious when it comes to that. So I would say only with permission from somebody like you, Don, in the regulatory branch, and as an only resort, you know, if there's no other option available to you. That's what I would say.
No, it's essentially what I said.
Yeah. Okay. Well, no, that's good to know. But yeah, I'd never heard of that before about importing foundation, wax foundation overseas. But that kind of straddles the line of kind of biological material, right? You're certainly not allowed to import live bees, live queens, pretty much live anything. But
Yeah,
beeswax, it is a medium that, you know, can harbor some live things, but I think it's not, certainly not sterile, but I
not a
don't
high
think it's
risk,
a,
but
yeah,
not
exactly,
not no risk. Yeah.
exactly.
Oh,
I
thank you,
definitely,
David, for all the product that you introduced. You haven't talked about venom, that
ah,
in some countries,
yep.
people are harvesting venom as well. So you may talk about that, I mean, brief if you wish to.
Yeah, no, I was going to include it, but for time I kept it out. But you're right, Eshmel, that there is a way to harvest without killing the bees, by the way, to harvest venom from beehives, and then it gets dried down into a crystalline form, and then you can sell that. And that can be really, really helpful for people who are going through immunotherapy, who, you know, are trying to get desensitized to honeybee stings. And so, you know, biopharmaceutical companies are are uh, is a good market for that. Although I don't know if it's a huge market. But that's, you're right. That is another product. And then of course, just apitherapy by the beekeeper themselves, where they're just stinging themselves. But yeah, venom is yet another product that can be important. Why some beekeepers are keeping their bees.
I mean, if we have time, I would like to add a few more sentences because
Sure.
with all the climate changing and scarcity of the flowers, I think at least like sideliners, they are moving toward not the single product of honey, they are moving toward the multi-product of the colony.
Right.
That instead of only having one product as honey, they go to different product as a royal jelly or propolis or venom, and then they have different resources of income that may help in,
Yep.
you know.
Yep. There's also from Lindsay and Jean, they had the same comment, Eshmael, about the bee venom for therapy for arthritis and other things. So yeah, so apitherapy is yet another Hive product, if you will, that you can have the bees for and use them. It's harder to find the market, I guess, of the harvesting it and selling it, but you can certainly do it and use it for yourself. Exactly right. Well, with no further ado, I'm going to go ahead and introduce our special guest for the evening. Kaira Wagoner is a postdoctoral scientist at UNC Greensboro, and she recently got her PhD. Well, actually, yeah, several years ago, she got her PhD from the University of Greensboro, and so she's stayed within her alma mater there. Before that, she got her Master's of Science from Greensboro, studying mosquitoes. She got her PhD, obviously, studying in honeybees, and we'll certainly talk about that. She got her bachelor's at Guilford College. So she has been in Greensboro for quite a long time. And she has done some really excellent work in her PhD and beyond on brood signaling and hygienic behavior as a way of bees helping to reduce disease within the colony. And so I want to thank you for coming, Kaira, and for being our guest this evening.
Absolutely. Thanks for having me.
Yeah.
Always happy to talk about bees.
Yeah. So it's always great to have somebody local as well talk about what they're doing and to showcase some of your work. So I have a quick slide here that you shared with me of that kind of summarizes a little bit of some of the things that you've been going on and testing a potential product to test for hygienic behavior. So first, why don't you explain to us what hygienic behavior is and why it's useful for bee health?
Sure, absolutely. So hygienic behavior, I'm sure many of you have seen it or are familiar, but hygienic behavior is the ability of those adult nurse bees patrolling the brood comb to detect and remove unhealthy bees from the colony. So if there's a varroa mite or a virus infected individual, they can detect that even through the cap of a capped brood cell. And then they can uncap that cell, pull that unhealthy brood out of the colony. And that small sacrifice, it is a sacrifice, but that sacrifice improves the overall health of the colony. So it's a really important behavior, and it's actually been selected for by breeders for several decades now. And there are some studies that show that hygienic colonies have less disease. So things like chalk brood can be managed pretty well with hygienic colonies. But the ability of hygienic bees to deal with varroa is a little bit more iffy, I'll say. And so what I did with my PhD, what I was working on is chemical communication. So I'm really interested in pheromones, chemical communication, especially as it relates to hygienic behavior. And right now there are a couple different ways of selecting for hygiene. So some of the most common are like removal of freeze-killed brood, the freeze-killed brood assay developed by Marla Spivak, and removal of varroa infested brood itself. But these selection methods that exist now and that breeders tend to use, they tend to trade off efficacy for efficiency. So the selection methods that are pretty easy to use, like the freeze kill brood assay, I would say is a little bit faster in terms of labor input. You might get some disease resistant bees out of it, but whether your bees will actually be resistant to mites is another question. On the other hand, you can select for suppression of mite reproduction in a cell, but that's really labor intensive. You get great bees that are very good at removing varroa, but it's really labor intensive. So what we're trying to do now is use these pheromones that we've identified on brood that are either varroa infested or deformed wing virus infected. And we've found that these certain pheromones are actually elevated in those unhealthy bees. So we've known for a long time hygienic bees are better at detecting, an unhealthy cell. But my question with my PhD is, what are they detecting? So there's something going on inside that cell, something that's coming out that they're smelling. And so we identified some of those chemicals.
Yeah, so you got those chemicals, and then you've done, so this slide here, it shows this test that you have of actually spraying these chemicals on sections of contiguous capped brood, and then coming back and measuring hygienic behavior later. So tell us a little bit about that.
Exactly. So we've taken a mixture of these pheromones that are associated with the unhealthy brood, and what we can do is apply them to just a small section of capped brood comb, and we're basically just spraying this liquid onto the surface. It evaporates really quickly, at least the part you can see. You stick it back into the colony for 2 hours. So it's a really rapid test. You stick it back into the colony for 2 hours and you pull it out and look at what the bees have done within that 2 hours. So in the top right here, you can see that all that uncapping in the high, we call it the UBO, or unhealthy brood odor assay. So you can see that there's a whole lot of uncapping there on the top picture. And in the bottom picture, you can see the bees have not done very much. And so what we found is using this pheromone assay, we can do like a June test on our colony. And with that one test, we can predict varroa mite infestation for August. So how they perform in June tells us a lot about how varroa resistant that colony is.
Now, remind us of how long it takes to do the freeze killed assay, and why, you know, a 2-hour bioassay might be superior to that.
Sure. So the freeze kill brood assay, it's a 24-hour wait. So, you know, you don't actually spend a whole lot of time doing it. I would say the hardest thing about the freeze killed brood assay is getting your hands on liquid nitrogen.
Yep.
Right. Getting liquid nitrogen is the tricky part.
Which we all have doers of in our garages, I'm sure. Right.
Right.
But, you know, yeah.
Yeah, even I am not a huge fan of working with it, although it's been a really useful assay to use in terms of research. But the time is 24 hours. So I know, you know, with smaller scale beekeepers, it's not such a big deal to wait 24 hours. But I think in commercial settings, it's a big difference to be able to just know right away what your colony's up to. And I think, you know, what we see is that with freeze kill brood, you don't necessarily have an accurate prediction of varroa resistance.
Right.
And what we see is with this pheromone assay, we can get a picture of how resistant your colony is to varroa. So there's lots of really cool things you can do with that information.
Well, that's a really exciting nuance there, right? That it's not just of how good the bees are able to help themselves. It's also an indication of how important you as the beekeeper need to take your mite control and prevention strategies
Right.
for each individual colony, right? So, you know, this is that time of year. So you've actually opened a business that is trying to bring this to market. Tell us a little bit about that.
Yeah, so it's a startup, so we actually don't have a product yet. You know, we've just kind of started the process. It's all based on this research. But the idea is to be able to bring this tool to beekeepers. So, you know, I've spent a lot of time actually working before my master's and PhD with ceramic water filters. In many different countries, and the focus is always trying to be simple, affordable, and effective. And so I kind of have brought the lessons I learned from that into my beekeeping world. And what we're hoping to do is provide a tool that beekeepers can use that's simple, it's not expensive, it's not hard to do, and it's really effective at showing you how varroa resistant the colony is. And there's all kinds of things you can do with that information. So some beekeepers may want to, you know, if you lose a queen, it's really valuable to know, okay, where do I want to pull eggs from? You can take eggs then from your most hygienic colony. You know, larger scale beekeepers can isolate maybe their varroa resistant colonies in another apiary, so you don't get as much drift of varroa into those colonies. And you can maybe do all your treatments in one yard and not in another. So we're really excited about all the potential applications of the assay.
Yeah, no, that it is really exciting. It's been making a big splash in the scientific literature, but as you said, I think it has one of these things that has such obvious practical applications that it's really great to see you pursuing that and hopefully getting some traction on that, because I know it's not easy to have a startup like that and to get all of the information, all the data behind a product like that. It takes a long time for these kind of things to come to market. So, you know, let's hope that momentum continues. I'm going to open it up to those who are watching on our YouTube channel. Again, thanks everybody for joining. Feel free to type in your chats ask your questions. There is a little bit of a time lag between us on the Zoom call and the YouTube channel. So we're just going to continue that conversation about hygienic behavior and bee health with Dr. Wagoner here. But feel free to type in your questions if you have any. But Kaira, I think you had mentioned that this is very helpful for varroa mites, which of course is kind of public enemy number 1, 2, and 3 for beekeepers. But is it also, do you think, helpful for brood diseases and some of the other issues that bees are dealing with?
Great question. I collected some samples today that are going to answer that question.
How about that?
Yeah, so I mean, I would assume that I think it's safe to say that if you're controlling varroa, you're controlling many different viruses. But there's a lot of deformed wing virus out there. There's different variants. And so that's kind of our next question is how well can this assay predict disease resistance? And does it do as good of a job as those other selection methods that are out there? So that's one thing we definitely want to find out. And actually Ismail, who's on the call here, is going to be doing some of the virus analysis for us for that project. So I hope to have a great answer for that in the next, let's say 3 months.
Oh, well, that's very exciting and very timely. Kirby here is asking about how they might be able to get access to the assay. From what I understand, it's not a commercially available product, but what can beekeepers do to help or to, to be able to take advantage of your findings thus far?
Well, thanks for that question. So right now, the main thing that's preventing us from having a product to get to beekeepers is actually our delivery system. So we use a solvent that's kind of dissolving those pheromones and helps spread them around that capped brood area. That's not very it doesn't get along well with plastic, let's say that. And so we're trying to actually just come up with a good way to, I have a system that works for research, but it has no shelf life. So I'm working on that. I have a couple companies that I'm talking to where we're trying to get just a good dispenser, basically. And once we get that done, I mean, it's pretty much ready to go. We're already, we're learning as we go. There's always more science coming out to inform what we're doing. So we already have identified some additional chemicals that we want to try mixtures with those pheromones as well. But we're hoping by next summer that we'll have something out that beekeepers can use.
Oh, that's really exciting. That's really exciting. Yeah, and I didn't do very well in my organic chemistry or beyond, and so I know how complicated these things can be for shelf life and things breaking down and the stability and all of that. It's in and of itself, it's a whole science behind the biology, right? So it's really quite a lot. But hopefully, if you're only kind of months or years away, that's really, really positive.
Right. And lucky for me, I have a great business partner, Phoebe Snyder, who, she handles a lot of the business stuff. And my focus is the research and the science. But together, that's a great combination.
Yeah. That's excellent. That's excellent. Richard asks another question, very interesting question here about, well, his comment is, do you find that Russian bees in general have greater hygienic behavior? But I'll kind of expand that out and talk about the different stocks of bees, right? You got Russian bees, you got Italian bees, you got Carniolan, you got, other types that are out there. What's the diversity of these different bees, different stocks, different breeders for hygienic behavior?
Sure. So I actually just tested my first Russians, and we tend to see with this assay, if you're familiar with the freeze kill brood assay, it has a threshold of 95% or more. So if the bees remove 95% or more of that killed brood within 24 hours, they're hygienic. With our assay, it's about 60%. Is what we find. And we had Russians that scored, I tested only 3 colonies this year, but they were all 3 just below that threshold. So I'm really interested to see how they do with mites, but they're close. So I think it's borderline. I tend to see that the VSH and the pole line out of the USDA, they tend to score really high. Hygienics can score high. The Minnesota Hygienics can score high on the assay, some of them lower. I just had, let's see, ran an experiment with 24 unselected colonies, so just your typical Italian colonies, and one scored hygienic out of the 24. So it wasn't sold as a hygienic colony, but I did have one that tested hygienic out of those 24, and it had no mites when I checked just a week ago in my 300 bee sample. So
Right.
so there is a lot of diversity, and I think that's one of the interesting things that beekeepers can do with this assay is use it as not only a tool to test their colony and make management decisions, but if you put money into a hygienic queen, it'd be nice to know, is it hygienic and how hygienic is it? And so
Yeah.
it can also, I think, be used as a tool for queen quality analysis and checking to see what kind of queen you have.
Yeah, validation I think is really good, not just for the consumer, right, but for the breeders themselves. They want to make sure that what they're selling is what they say it is. So yeah, no,
Exactly.
that's really great. Yeah, no, keep the questions coming, guys. This is really great. I really appreciate the questions coming in. I think the one thing that's always puzzled me, and there's one thing that we really need to keep in mind about hygienic behavior, is that it's a recessive trait, right? So that's why it's not just everywhere. You were mentioning about how, well, it crops up every once in a while in unselected bees. Well, you see that in recessive traits, but that's why Russian bees and some of these others that have been selectively bred for this or other behaviors, they're a closed population, right? So you're able to retain those recessive traits. But the second that you buy one of these queens, the daughter queens then mate with whatever drones that are around, you then lose this hygienic trait, right?
Exactly.
So, is there any advice for your average beekeeper about what to do once you have some of these hygienic traits in your colonies and you're seeing benefits, how to keep them around?
Yeah, convince your friends to buy hygienics too. I would say, at least your neighbors.
Right.
Yeah, well, we're running an experiment right now, actually, where we're taking sister queens, and we have a hygienic apiary and a non-hygienic apiary. And so we're taking sister queens from each of those apiaries. We made splits, and then we're requeening them in the two different yards. And we're going to see how hygienic they test compared to their mothers. So I think that'll be really interesting. I know it's tough to keep those genetics around, but it'll be really telling to see how much of that hygienic trait kind of stays with that daughter queen. But I think, yeah, it's a tricky thing. I guess in terms of tips, try to get your hygienic bees, if you have them, to produce a lot of drones. And keep new genetics coming in, but get them to produce as many drones as possible. So, if you can flood your congregation areas for those drones with hygienic drones, then you're going to have a better shot.
A better shot, but not a guarantee, right? It's another example of how beekeeping is always a heavy lift. It's active citizenry, right? It's not just fire and forget.
Absolutely.
It's a constant thing. Yeah.
Never a guarantee. Yeah. It's
right.
Always, there's one interesting thing I saw from a Greek group called the Virgin Queen Train. And what they do to try and control their mating is they keep basically the virgin queens and the drones locked up, because I think they tend to mate in the morning, and they release them then, just the drones they want and the queens they want to mate in the evening. And so I think there's some timing things you can do to try and manipulate. But I don't know how, from personal experience or research, I don't know how successful those strategies are.
Yeah. Well, whatever it is, it's not easy. But any tool that can make things just a little bit easier, a little bit faster, a little bit more reliable for the beekeeper is beneficial. And Kaira,
Absolutely.
I think that this product and this paradigm of research that you've been working on for 6-7 years now, right? It's really
10.
started. Okay, 10. It's really coming to fruition, and I really think it's great. And so we are really happy that you're part of the North Carolina beekeeping community, and just wanted to make sure that you had this platform for folks to know of your work and to support you in any way that we can. So,
Thanks so much. And I'm happy
yeah.
to answer any questions in the future too, if folks come up with them later. I love talking
Yeah.
about bees, and we're really excited about the project and where it's going. So I'm happy to talk more.
Well, if you don't mind, is it okay for me to send out your email or put the email on the chat here so that people
Absolutely.
can? Okay, I will do that. There it is. So again, feel free to email Dr. Wagoner about this at your leisure.
Look
So
forward to hearing from you.
Terrific. Well, before we end here, I just want to once again remind you that we're doing these apiculture onlines at the last Wednesday of every month. So we're going to a monthly format here. So our next apiculture online is going to be on August 25th. And as I said before, it's going to be a topic all about varroa mites, because July and August are the kind of most critical times of year in the Northern Hemisphere, at least, for managing varroa mites. And so we're going to have a very special guest, Dr. Gloria DeGrande Hoffman, who's the research leader of the USDA Agricultural Research Service Bee Lab in Tucson, Arizona. So she's going to be joining us from the West Coast and talking about varroa mites. She's done a lot over her very long career on varroa mite population dynamics and their control and overall bee health. So we're very fortunate to have her. So once again, thanks again for joining everybody who's on the YouTube channel. We want to make sure that you subscribe to our channel, like and comment on our YouTube channel, and we'll continue these webinars and the recordings as they come along. So once again, thank you everybody, and hope you have a good evening.